Everyone, I'm Professor Sarah Rankin and I'd like to welcome you to Imperial's COVID-19 lockdown lessons. Today is lesson 7 and we're going to be hearing from Professor Peter Openshaw and Dr. Ryan Thwaites, who are going to be talking about some of the COVID-19 clinical research and clinical trials. Professor Peter Openshaw is a world-leading clinical researcher into influenza infections. He originally trained as a doctor and has been a respiratory consultant for many years, in addition to doing the scientific clinical research. Throughout the pandemic, he has been leading national projects, coordinating and analysing COVID-19 patient samples and establishing some of the ongoing clinical trials. Ryan, on the other hand, has actually been one of the people that's been at the bench analysing the patient samples. So as always, Margarita is with us and she is going to be looking at the chat in YouTube. So please post any questions you have for Peter or Ryan as we go along. So Ryan, would you like to kick this off maybe by telling us how it was you went from studying biochemistry to working in Peter's lab?
Yes, thanks. Thanks, Sarah. Afternoon, everyone. Thanks for joining us. So I started my career in science working, doing my undergraduate in biochemistry. So I studied biochemistry, finishing in 2010. After that, I didn't really know if I wanted to work in the lab full time. And I felt like I didn't have enough lab experience at that point to really make that decision. So I found a job in industry for 2 years and worked doing that. Decided that I really loved working in the lab practically, loved science, and loved being able to think up my own ideas and coming up with solutions to problems. So then I did my PhD, finishing in 2015. That was in autoimmunity, and then decided to make the move into looking at kind of the immune response in infectious diseases, and was lucky to find a postdoc working with Peter. So really, to all of the students listening, one of the first things that I wanted to say was that there's lots of different routes and lots of different career paths in science. These are very varied and everyone has a unique and totally independent route to success. But there's lots of great career paths, whether you go down a very traditional academic route like I have to do postdoc studies, or whether you work in industry, or whether you take on some other completely different work. People quite often decide to change course during different stages of their career. So you might take in an undergraduate degree. For example, I did biochemistry, but then decided to move into immunology. And there's lots of flexibility within those pathways. You can have one goal at the start, but being flexible and kind of moving that aim, that motivation, is a perfectly reasonable thing to do. One of the unique things in science is that because we all have these different experiences, different undergraduate degrees, working in different labs, we have really different experiences that make us as a team stronger. And inevitably science comes with some setbacks. Sometimes your experiment doesn't work, sometimes your idea doesn't work. And those are perfectly normal and perfectly straightforward things that all of us have to tackle. But they make us stronger, they make us better scientists in the long run.
So Ryan, have you moved the slides forward?
I thought I had.
Right, because we're just seeing the opening slide.
No problem. Let me see why that is happening.
So Ryan said that he was lucky to work in my lab. I'd say that I'm lucky to have Ryan.
Oh, I like that. So that maze is people coming in from all directions into the Holy Grail, which is Peter's lab in the middle.
Well, either we can look at it as people all taking different routes into the maze to reach one point, or as all starting in the same place, i.e. we've all done levels,
yeah,
and we all come out to different points. So you can kind of look at it either way around. So yeah, this slide should have been up whilst I was talking just then. But I think the important thing to say is that there's lots of routes. And as I say, we either start from different places and reach the same point, or we start from a very similar place and end up in different places in life. And that's all part of the fun.
So tell me, just why did you choose biochemistry in the first place?
I was really torn between biology and chemistry. I knew that I wanted to do something that had impact on health and kind of studies with humans, but I didn't know whether that might be chemistry and going into pharmacy perhaps, or really biology. And basically I couldn't decide. So I spoke to my chemistry teacher and he suggested, after several hours of being frustrated with me, that I should just apply for biochemistry. And it was probably some of the best advice I got. I really enjoyed my course, and it had enough flexibility in it that when I finished, I decided I definitely didn't want to do chemistry. I wanted to do slightly more human biology work and started progressing into immunology.
Okay.
So, should we move on to the kind of coronavirus piece? So I'm going to hand over to Peter to talk through the first of our slides, actually about COVID and the work that we're doing with COVID at Imperial.
Okay, yeah, so maybe I should just say, I mean, I started out really not knowing what I wanted to do. I was just interested in so many different things, but I was fundamentally interested in how people worked, I think. And I was also, you know, a bit of a Boy Scout. I liked mechanics. So my first degree was actually in lung mechanics, you know, how things stretch and break and, you know, that sort of stuff. So, but yeah, so it's interesting how you get into your careers. So just a bit about coronavirus. So, you know, we've all been infected by many coronaviruses of the common cold variety. There are about 4, you know, well-known coronaviruses. And because they mutate at a quite regular way, you know, their genome is a bit like a molecular clock. You can work out how long it's been ticking since it started back way when it was in another species. And in that way, you can try to estimate how long a virus has been evolving in a set species, how related it is to other species. And so with this coronavirus at the top left, that's called NL63. You know, it's been estimated that that's been in the human evolution for about half a millennium, whereas the HKU1 only jumped into human evolution back in the early 1950s. So it's quite well related to animal coronaviruses. And then we've got SARS-CoV and MERS-CoV, and now the new SARS, the updated SARS-CoV-2, and those we've been able to actually witness in real time, their emergence and their the formation of a new pandemic. So just comparing, you know, SARS 2002, which I remember very well. I was working on various advisory committees and working with viral immunology back in those days. And comparing it to the COVID-19, you know, I think the thing which really was in our favor in 2002 was that actually it was quite hard to transmit. Most of the transmission was going on in enclosed environments, particularly in hospitals, but sometimes in hotels, and people were quite symptomatic when they transmitted. It was very scary. You know, we all thought this was going to be a pandemic, and it jumped to Toronto and nearly did, but eventually it was defeated by public health measures. COVID-19, bit more infectious, more variable in severity, you know, some really nasty disease, but quite a lot of mild disease. And this one just managed to squeak through and escape containment. And we can see that on the next slide. You know, I think the Chinese were actually doing really well. They'd almost managed to lock this one down. The case numbers were really, really dwindling, and they'd warned us all that this was a nasty one and that we had to take rapid action. But sometime in late February, I remember hearing that it had spread to Iran and to Italy. And at that point, I thought we're lost. You know, there's no way that we're going to be able to do what the Chinese did. And that's actually what happened. It really exploded from there, became a European disease, and now a Pan-American disease. So yeah, so back to Ryan.
Great. So I'm going to talk through some of the literature, some of the research papers that have already published around COVID-19, and then go on to talk a little bit about what Peter and I are doing in the studies that we're conducting at Imperial. So the paper that I'm showing here was published really quite early on. in about in about April, which was looking at the relationship between the amount of virus in the airway, which we call viral load, and how long people had had symptoms. So on the left-hand side of this plot, we can see that quite early on in symptoms, so perhaps within the first week or so of symptoms, people have really quite high viral loads in both the nose and in the throat, which is where we get these diagnostic swabs from for COVID. So particularly in the first week, we have really quite high viral loads, and these decline fairly predictably in most people through the kind of second week of infection, to the fact that most people no longer shed virus that can be detected within about 3 weeks of symptoms.
So Ryan, can you just tell me that on that chart is each one of those dots is a person?
Each one of those dots is a single person over time. Yeah, so this is a kind of an accumulation of data from lots of different people, lots of different patients, and plotted against what they reported to be the start of their symptoms. So, yeah.
And I think also for students, this is the type of trick question you get in your sort of A level, because it's really important to note that you've got a log scale going up the y-axis, and so it doesn't look like a really dramatic reduction, but when you consider it's a log scale, it is quite a dramatic reduction in the titer. Yeah,
absolutely. So really in the first week we're talking about hundreds of millions of copies of the viral genome being detected in the swab, and that falls down to perhaps a few hundred in the later stages, but it's still detectable. But absolutely, yeah, it is almost a trick question set up. So the plot on the left shows viral data that's measured by looking at the copies of the viral genome, copies of its RNA, which make up the genetic material for the virus. But that doesn't necessarily mean that the person who might have had symptoms for several weeks is still infectious, because those viral RNA copies might not actually represent an infectious virus. So one of the things that people also have to do is to try and culture the virus from those swabs. So that means that you take your viral swab and you inoculate it into a culture of cells that can be infected with the virus, and then see whether you get this positive culture on the right-hand side here. And when we do these tests, we see that actually most people are only infectious within the first maybe week or so of symptoms with COVID. After that first week of symptoms, most people become culture negative, meaning that we can no longer get live virus out of the swabs. And these people also start to produce antibodies around this time as well, which is in the top plots of this right-hand graph. So this shows that as people start to mount an immune response and generate antibodies, they also start to clear the virus, and particularly start to clear live virus from the airway. But you might still get shedding of viral genetic material, viral RNA, which you pick up on these diagnostic swabs. So if we take all of this data, accumulate it with lots of other different studies that are going on and lots of kind of small studies that are coming out around the world, we start to build up a picture of what COVID-19 looks like and the different phases of the immune response. And this is really where our interest is. So if we start on the left-hand side of this big blue arrow here, we see that around the time of first symptom onset, you've got really high viral load in the airway. This declines fairly predictably through the first week or so, whilst you're maximally infectious. So at that time, when you're shedding lots of live culturable virus, you're more infective to other people. But after the first week or so, this virus load starts to decline. You start to see antibodies appearing. But some people still progress into kind of increasingly severe disease, despite the fact that the amount of virus really seems to be diminishing. And we think that this progression into more severe disease is actually associated with an inflammatory phase of the immune response. So while the virus might be being cleared from the airway, the immune system takes over, and you perhaps start to get this kind of immunopathogenesis, where the immune system starts to contribute to tissue damage, which leads to increasingly severe symptoms, particularly in people that are at high risk. This inflammatory phase can continue for a few days perhaps and might resolve, or can continue for several weeks, particularly in people with very severe disease who might need ventilation on an intensive care unit, or who might sadly die from COVID disease.
So when you say shedding, you know, that sort of conjures up a picture of sort of us shedding our skin, but you mean through sort of coughing up, and that's how you're sort of getting rid of the virus?
That's right. So when we talk about viral shedding, we kind of mean all of the droplets that maybe come out when you sneeze. That could be viral shedding, or when you cough with COVID, particularly little droplets of viral containing fluid come from the airway, come from lower in the airway, and that virus is shed either into the environment or just represents the amount of viral replication that's going on in the airway. So really, shedding is very similar to viral load for measuring it immunologically, but it's also important from how infectious a person is, from them shedding the amount of virus that they shed into the airway or into the environment around other people.
Yeah, but maybe I could add to that and say that people often in their manuscripts, they write viral shedding as if to imply that that is, you know, spreading it around and infecting other people, whereas actually what they've measured is that PCR detection of the virus's RNA, which doesn't actually mean virus shedding at all. You know, because as you just said, a lot of that that you can detect is probably just degenerate genome or virus which is on the way out. It doesn't mean to say that it's actually infectious. So I think it's a bit of a misused term. I think.
Yeah, and in the same way. So when I remember sort of early on, we were being told that the virus could stay alive on surfaces for up to 3 weeks. Is that, and that was by measuring sort of again by PCR. So are we any further in knowing whether that virus is live and what surfaces are good or bad or anything?
I don't think that's infectious. I mean, you know, it actually is, it's an RNA virus. RNA is quite unstable typically, and is very easily damaged by UV light or by heat or by, you know, disinfecting agents. You know, I think almost all the transmission, to my mind, is happening with face-to-face exchange of droplets, which are either sneezed or breathed, or, you know, droplets which are expelled whilst you're talking or shouting or singing or, you know, greeting friends. You know, it's that sort of thing which is really high risk. And I think picking it up.
So do you think, I mean, so I know people that are keeping Amazon parcels in, you know, separate rooms for 3 weeks before they open them, or when they're getting their shopping in, they're using wipes to disinfect every package. Do you think that's
almost no evidence that that is a mode of transmission? I think the problem is that some people are spreading the virus, live virus, when they don't know they've got it. That may be because they haven't got symptoms yet, so they're pre-symptomatic, or they've only got a mild, you know, aches and pains. They're not feeling 100%, but they're still okay to go out and socialize, and particularly to go to that, you know, longed for birthday party at which they hug and sing and chant and dance around. You know, don't want to miss out on that. So you go, even though you're feeling a little bit rough, maybe take some paracetamol to get you through the day, you know, and that's really high risk. So I think, you know, a parcel, even if it's come from a place that you think is a high prevalence, you know, I honestly don't know of any examples of people catching it that way. Okay. It's all through this direct contact with droplets, and particularly droplets of saliva and mucus.
Okay, cool.
So I'll move on to talking slightly about the work that Peter and I are doing under a study called ISTERIC. So ISTERIC is a UK-wide consortium that was set up following the flu pandemic of 2009. This flu pandemic wasn't anything like the COVID-19 pandemic, but was really seriously worrying, at least at the start, when we had no idea how severe this virus was going to be. So Mosaic, which was the flu pandemic study, designed a clinical data tool, which is used in all of the studies, all of the centres participating in ISTERIC. which allows all different hospitals to collect the same information on their patients with COVID-19. And this is really important because it allows us all to pool data between all these different hospitals that are caring for patients with COVID-19 in the same way, meaning that we can look at data really comparably between all these sites. The study that we're talking about
so I'm just going to butt in there because I know there's something that comes up a lot in GCSE and A-level science, and this idea of how you have controlled experiments and how you have to keep all the sort of different conditions, you know, the same, and you just want to look at the one that's changing. And so this is very similar to that. It's trying to sort of, as I say, make sure we have all the right data, because if you want to find out, is it more prevalent, say, in women than men? Is it, you know, the recent studies on the sort of BAME population and trying to dissect out that, the more information you gather, the more easy it is to interpret the data. So tell me one thing, what does ISARIC stand for? Some long...
So it's a bit of a mouthful. ISARIC is the International Severe Acute and Emerging Respiratory Virus
Consortium.
Infections Consortium.
And how many people are part of that? How is it across Europe or across the UK, or how does it go? How does it work?
So ISARIC really is open worldwide, actually. So
okay.
Places around the world can use the tools that we have generated under ISARIC, particularly the clinical data tools. This has been translated into, I think, more than 50 languages to be used around the world. So the idea is that centres worldwide can collect the same clinical data about their patients as we are doing in the UK. Mm-hmm. The majority of cases that we're going to talk about are from the UK, but there are groups around the world that are collecting this data as well to be included in the study. So
yeah, so presumably at the moment you're analysing the UK, you're looking at the national picture, but in the future or later down the line, you're going to be combining that data, and then you can compare. Yeah, because that's going to be really interesting to see, you know, how it sort of everything panned out in different countries and why. Yeah,
absolutely. Yeah, it's such an important question, and to be able to answer those questions in the years to come, we need to have done the groundwork now to make sure that all the data is the same, that it really is a controlled experiment, as you say. So that's one of the really important things that ISARIC has done. I'll quickly move on to what ISARIC has done really since the pandemic started. So this is a summary of all of the patients that ISARIC has recruited, which I got from our website yesterday. So in total, we've recruited more than 66,000 patients to gather their clinical data. So this first tier, tier 0, is really that clinical data form that I was talking about, where all hospitals have completed the same form for each patient. This looks at their demographic data. So we mentioned ethnicity. It looks at things like ethnicity, sex, age, and any comorbidities that you had. So things like chronic lung conditions or diabetes. That's 64,000 patients. Then we have 2 tiers of sampling where we look at the biological sample collections as well, which total more than 2,000 patients. So those have had blood samples collected, nasal swabs, or other respiratory samples, as well as urine and stool and a manner of other biological samples. So I was speaking to our collaborators who kind of manage the collection of these samples last week, and they've had more than 45,000 biological samples collected from these patients since January or February, since we started. So that's really a huge concerted effort from people around the UK.
And I think it's also really important to mention that, you know, that it's obviously, you know, difficult when patients are coming in with an illness to sort of sign up to this, but it is really important for us to then be able to work out exactly what's going on. And I think some people are sometimes, you know, they don't understand why, you know, why are you asking me about my ethnicity or something like that? What's that got to do with this? And so, but I think yeah, I think the results speak for themselves, don't they? Or the results that have come out. But that is why you're being asked all these questions.
Yeah, I think it's another thing to say about this is it's amazing to see this happening. You know, we sort of tried to do this in 2009, and it took us so long to really get going that most of the cases had already been and gone by the time we were set up ready to go. So ISRIC meant that we could launch immediately, capture patients from day one. And I don't think there's any other country in the world that has such a coordinated healthcare system that would allow this study to be done. You know, we've captured 45% of all the UK admissions to hospital with COVID. And, you know, tribute to the NHS and to the literally thousands of people who've been involved in putting patients into the study. It's amazing.
Yeah, and the other really important thing is, I think we've mentioned this in some of the other talks, that if you're going to do a study like this, if you're going to do a clinical study and you want to collect patient samples, then you obviously have to get ethical approval and you have to have patient consent. But the design of the study and what you intend to do has to go through a number of different ethical boards, and especially something where you're going to get lots of different countries involved. So it would have to pass all, you know, for example, the European Union's ethical boards, etc. And so that would normally take, you know, could take between 6 and 9 months to actually get ethics approval for a study like this. And that was why it was that forethought and making sure that this was all in place for the next time, which is now, that you've been able to do this so successfully. So it's really a credit to having things in place.
Absolutely, and it speaks testament to the need for planning in science, that so much of what we do on a very small or a very big scale really comes down to meticulous planning. And this planning was 10 years before it really was needed, but boy, is it useful now.
Yeah.
So the first outputs, really, the first research paper that we've had from ISRIC is one that was published a few weeks ago in the British Medical Journal. So this looks at data from more than 20,000 COVID cases around the UK that were hospitalised and treated in hospitals, in 208 hospitals around the UK. And as we mentioned earlier, the number of people involved in this, in terms of approaching the patients or their families, consenting them to be involved in the study, collecting research samples, analysing those samples, comes to 2,648 people just in the frontline NHS setting. And remember that this is really during the peak of that first wave of the pandemic, where there was really acute need in the clinical setting. People, there was lots of patients coming in every day, and to be able to recruit and approach these patients required a huge army of people, and it's a fantastic achievement for everyone that's been involved. And it's, as Peter said, it's by a long way the largest study of its kind worldwide. So this recruited 20,000, just over 20,000 patients. At the time, that was 34% of new cases in the UK. As Peter said, we're now at about 45% of all COVID cases in the UK. And this was really looking at predictors of severity. So whether a patient was admitted to ICU, whether they died, based on their clinical characteristics. Peter is one of the leads for this project down here in the bottom right, along with Callum Semple and Kenny Bailey. And this paper was published by Anne-Marie Doherty as the first author. So these people are from Liverpool, from London with us, and from Edinburgh. So it's a kind of UK-wide consortium team effort that has really led to being able to do this so well and so quickly. So I wanted to talk for a minute or so about what I do and what my kind of contribution to ISRIC is. And I'm a lab scientist predominantly. So a lot of what I do is on the left-hand side, which is a pipette. I spend lots of time moving what look like colourless liquids into other colourless liquids, and then trying to measure them in different ways. The thing in the centre is an ELISA plate, which we use to quantify or measure the amount of different proteins in maybe the circulation or in the respiratory tract that might tell us something about the disease and how the disease is manifesting in different patients. And then on the right-hand side, we've got a computer, because all of us that work in immunology or work in clinical science have so much data. You can imagine that with 60,000 COVID cases, there is an enormous amount of clinical data, as well as things like biological measurements that we need to combine in order to understand how different factors contribute to severity of these cases. So computer science and the kind of an ability to work well with computers is increasingly important, as well as all of these kind of bench skills or laboratory skills that you might be more familiar with from kind of GCSE and A-level sciences.
So I'm just looking at your ELISA plate there, Ryan, and I'm guessing, well, the first thing I was going to comment again for all of the students out there, remember when you do an experiment, you do everything, you don't do a single test, you always do multiple tests. So each patient sample, I am guessing, Ryan, you've done these in triplicate.
Yep.
Because I can see there's groups of 3 samples that have the similar colour, and where you're pointing there on the left, that looks like your standard control that's going to generate your standard curve. Yeah, is that correct?
Absolutely. Yep, got it right. So on the left-hand side here, we have the standard curve, which goes from a very high concentration of something that we know the amount of, through a serial dilution, where we decrease in a known way the amount of what we're putting in. So this acts as our control for the experiment, where we know how much of a certain protein, for example, we put into the experiment, and then it gives us a color. We can then generate a standard curve based on these controls and compare all of our unknown patient samples, where some of them have very high amounts and some of them have very low amounts of these proteins. And then we can compare all of these data to other things about those patients, for example, their severity.
Yeah, great.
So really, one of the key take-homes, I think, from what I wanted to say was that science is a really big team project. Whether that means working in a team with people in your own laboratory, where you might have 4 or 5 people that you work with on a day-to-day basis, in your department, in your university, or whether that's kind of global and worldwide, just from publishing research that other people read and use to inspire their own work. So science is also a very creative process, and quite often people talk about scientists as being very dogmatic, very set in their processes, but actually science allows you to really think outside of the box, to try and do your reading, understand your topic, and then come up with new and really novel ways of trying to solve a problem. And that's a fantastic thing. It's one of the most enjoyable things about jobs in science, that you get to think about problems, really detailed problems, and come up with new creative ways of solving them. So in our example, in ISRIC, we have a cycle of what we do, where we collect samples from patients admitted to hospitals. I'm then involved in analysing those samples, which feeds into the need to communicate our findings. So we talk to other scientists, we talk to the government, we talk to newspapers, to anyone that we think would be useful to tell the things that we find. This then leads to us, along with the rest of our peers, considering opportunities for new treatments and collaborating with other groups who conduct clinical trials to try and inform those new treatments, and then work backwards again to collecting samples and seeing whether those treatments work. So communication is absolutely key, whether that's with the people you work with every day or through writing papers and talking to people at conferences. And you really have a long-term career in science, so you need to be kind and helpful and generous to work with other people. And you never know when you might meet someone that you saw again. So some of the people that I did my undergraduate with, or I worked with in industry, I see at conferences, and they're fantastic friends and collaborators. And you never know when you might need their help. As I mentioned, science is also a really creative process. One of the best things is being able to think up new ideas and test those ideas and see which ones are great and which ones are bad. So amongst all of the ideas that come falling out of our head at totally random times, sometimes in the middle of the night, sometimes when you're thinking about something totally different, there's always one wonderful idea in there in the middle. And you've got to try and pick which idea you think is the most important to test. And to do that, you talk to people, you share your findings, and you try and come up and improve your ideas. Sometimes your ideas seem very simple and very obvious. You think, why has no one done this? Surely this is the most simple, straightforward thing that we could possibly imagine. However, once you actually start testing your hypotheses, once you design your experiments, there can be various and many pitfalls that you fall into. But this is all part of the fun, really, that you get to discover those things that you need to work on, the reasons that other people might not have done your studies before. And ultimately, that all of that work makes hitting your final ends, finally reaching this point B up this mountain, worthwhile.
Great. Thank you, Ryan.
It's, yeah, I mean, it is really long-term science. You know, when I was doing clinical work, you know, you'd see somebody every 10 or 20 minutes, and in that time you'd listen to what they had to say, you'd analyze what it was, you'd talk to them about it, and you come up with a solution and you move on. So it was very quick fire. And science is slow burn, but it is hugely creative. So I'm actually married to an art historian, and I would say that, you know, in art history, they try to put aside any sort of judgmental pleasure these days that they might have in a picture, because they want to make it all very objective and measurable and all the rest of it. You know, in science, actually, we're allowed to be really creative and to find beauty both in ideas and in images. So it is a very, very creative process. Sorry, Sarah, I interrupted you.
No, totally agree with you there. I mean, I think I've been reading lots of sort of crime thrillers recently, and I think that's, as scientists, we're part sort of detective. You know, you're trying to look for clues and solve something, and we're also inventors and we're innovators, and yeah, using new technologies, seeing what more we can find out. Yeah, and I think it's that joy of sort of doing really elegant experiments and getting interesting results and finding out things that people never knew before. So Ryan, it must have been, I know you've been incredibly busy working hard during the lockdown, but it also, I'm sure it must have been really exciting, and especially knowing how important that work is. So, as you're aware, the government has needed to consult with scientists. We're hearing all the time that the government is consulting with scientists with respect to any sort of issue. So whether people should wear face masks, whether they need to be 1 or 2 meters apart, etc., etc. And in the last month or so, Peter has been one of those scientific advisors. So I just wanted to ask you, Peter, sort of how did that happen, and what does it involve, basically?
Yeah, well, it's very interesting, you know, extension of your role as a communicator, really. So I was a member of this committee called SAGE back in 2009 to 2012 or so. So, you know, I've been a government advisor going back to about 2005 in various guises. And, you know, sometimes I've been asked to consult by, you know, people at Porton Down in, you know, who are thinking about, you know, how to control outbreaks. It's very interesting to try and learn what sort of things people who are not in your field want to know about pandemics, about infections, about how respiratory viruses are transmitted. And it's really a matter of trying to use your judgment on every level to think, what is the key bit of science you want to communicate? How can you get it over in a way which is succinct and, you know, is within the attention span of your audience, whether it's a member of the cabinet or your peers. You know, you have to really try to adjust the style of delivery depending on what you're going to do. So I was chair of a committee called Nerve Tag at one stage. That's new and emerging pathogens, with advising government. So that's really the purpose of Nerve Tag. At the moment, I'm the vice chair of Nerve Tag. It's chaired by Peter Horby, who's in Oxford and is an infectious disease specialist. And what we do is we consider questions which come down from government, and we produce a distillation of all the science in a way that we think can be assimilated by those who need to know. So it has a big impact, what we say, if they listen to us, but they don't have to follow what we advise. You know, we advise and they decide. So it's the politicians who make the decisions about what will be followed and what won't, but we tell them what the science is. So it's a really interesting role. I think you just have to be calm if they don't follow your advice. You have to appreciate that they have other imperatives in addition to the science. But at least we have a system in the UK which is almost unique in that we've got these very complicated committees set up to produce advice, which is then fed to government, and potentially they can listen to it. I'm sorry there aren't more scientists actually in government. I think that would be really helpful.
Yeah, I was going to say that there are very few politicians that are scientists. And it's interesting why that is. I think we should encourage more scientists to go down that road.
Merkel is a good scientist. Yeah, Maggie Thatcher did science, didn't she?
Yeah. But yes, as you say, Angela Merkel's being a scientist, and so really interesting. So let's move on. I think we've got some questions. Margarita, are you there? And are you going to tell us some of these questions that have come up?
Yes, Sarah. Hi, thank you for this incredible database and consortium, if I can say that. I checked the webpage and it's really user-friendly and you can learn a lot of things from that. So I have some questions for you. Actually, one of our listeners posted 3 questions. 2 of them are more generic, has to do with something to be politically correct or not. And one of them is more scientific. So I'm going to start with the scientific one. So could you explain the thinking behind 7 days quarantine for symptomatic people, but the 14 days isolation for their household at the beginning of the crisis in relation to the viral setting mentioned?
Shall I take that one? So after you've developed symptoms, you become non-infectious after about 8 days, and probably by day 7 you're not very infectious at all. You know, you may still have PCR detectable bits of virus in your nasal secretions, but you're not actually very infectious. So, you know, that's a reasonable time to start coming out of isolation. People who live with you are going to take on an average of 5.2 days to become ill after you've infected them. So, and some of them will replicate the virus and will be capable of spreading it without having any symptoms. So by extending that up to 14 days, that means that you'll catch those asymptomatic spreaders as well as people who are going to develop symptoms, because you can have quite a long interval between exposure and actually showing symptoms. You know, the outside is about 12 to 14 days. So it gives a reasonable window for trying to protect. But it's a balance of judgments how long we should have. But 1 week and 2 weeks seemed an easy number. When we were asked, that's what we came up with.
Yeah, and the same for, that's why you have to go into quarantine for 2 weeks now after you've travelled, in case you've sort of come into contact with someone during the travel.
Yeah, but I think it's interesting to think of ways you might shorten that by doing more testing, for example. Yeah.
Okay. Yeah. Next question.
So I move on to the next question. Mm-hmm. How many different countries have you collaborated with for the first time during the crisis? Is there any international scientific relationship that has been strengthened in particular?
So I think in terms of the ISRIC collaboration, actually the kind of data tools and the clinical tools that we release are just open access. So studies can take those and use those locally and add in the data at a later point. So I don't know how many countries we're collecting data from at the moment, but it's continuing to grow and will continue to grow, I think. But on a kind of more general level, I'd say that the global collaboration of the scientific community as a whole during the pandemic has been really unparalleled almost. We've moved from a system where we would kind of keep most of our results to ourselves or within a small group of people that we trusted until we were really ready to release everything in one big paper. And we've moved from that system to really people releasing snapshots and little snippets of their understanding as it develops in a much quicker way to try and add small bits of new information that might help other people in different countries. So I think the kind of global collaboration really has changed a great deal during the course of the pandemic.
And we've also used similar platforms to this, like Zoom and other platforms, to host live seminars from experts all across the world. So we'll have, you know, respiratory people from Italy and New York and the UK sharing their experiences. And I think, again, that is something that is unique and related to the sort of you know, that remote working sort of ethos that we've all had to change to.
Yeah.
Yeah.
I
think it's one of the advantages.
So, I mean, when I was back in the 1980s, we were very sort of US focused, and that continued through the 90s. You know, we had a lot of US collaborations in that time. I used to spend a lot of time traveling to California in particular, which was great. And then with the rise of EU funding, we became much more European focused, and I've been in some fantastic European collaborations, discovering that Europe too has great scientists. You don't have to go to America to find them. And I do hope that all of that will continue. I can't see how science can be anything other than truly international. In most labs, you know, 50% or so of people come from overseas. And in the best labs, you find, you know, a higher proportion, I would say. So it is a really international thing. And I do hope that Brexit doesn't get in its way.
I'm glad to say that, because I am an international student, so I can understand what you mean. And the next question has to do with Brexit. And how will Brexit affect the scientific collaborations and the political changes, how they influence these open access databases? It's a general question. I think you can join me.
I think so. There's the UK at the moment, at least, is remaining part of the bigger EU science funding schemes. So under the Horizon scheme, the UK will remain a member. And I think that the funding landscape might not change so much, but as Peter said, what's more concerning is the change in landscape for people being able to move freely between labs in the UK and Germany or anywhere around Europe. And that's been something that we've really had available to us for my whole career, that we can come and go around Europe as we please. Now that's been extremely good for scientific research, as well as for the student body around the universities as well. So I do hope that those don't significantly change during Brexit.
Yeah, and I'm sure Peter has the same experience as me, in that you have people from, you know, different places in Europe come and work in your lab. When they leave, they go and establish their own groups in different parts in Europe, and you maintain that network of people over many years, and it sort of gets bigger and bigger. So you have a network you know, that is global because of there's a lot of moving of people between the labs. So in science, you do a lot of short-term contracts in the beginning of your career, and that's one of the great things about science. So like Margarita has come to the UK to do her master's degree. You can do your PhD in another country, which is another level of a degree when you get your doctor, and then you have up to 6 years when you can travel to different countries working in different labs. So as a scientist, you're in a very privileged position because you usually can get visas to go and work in different labs in different countries, and certainly obviously in Europe we've been allowed to do that exchange very easly. And it does mean that as we are a sort of real global community, I feel that's something that is quite different maybe about other careers. It is a very sort of a job where you are because if you discover something, then that's going to be considered by scientists all across the world, not just people in your lab or your university in the UK. What would you say, Peter?
Yeah, well, I was just thinking it's such a change in the last few months. I mean, I think that my last tally, I track my Google, timeline, and I think I've been to 50 something different countries and traveled many times around the world, in the pursuit of the perfect conference. And I'm actually, it's really exceptional for me to be here, so much in one place. I don't know what the future holds in terms of conferencing, but I do think going to conferences, meeting people all over the world is such an integral part of what we've always done.
But you must also have, if you sort of had a picture of the world and plotted where all the people from your lab are now, they would be spread across, wouldn't they? So you have contacts all across the world. Yeah. Great. So Margarita, is that it in terms of questions?
Yeah, we don't have any other questions.
Okay, so thank you, and thank you to those that asked questions. Just leaves me to say thank you to our speakers, Peter and Ryan, for those really excellent talks, really hot off the press in terms of what's going on at the moment in the UK in terms of the clinical research. And thank you again for Margarita for moderating the session, and Sam for producing the session. We will be back tomorrow at 2, and we're going to hear from Dr. Blarina Armetajashala and some of our pharmacology students, who will be telling us in more detail about some of the drugs that have been used to treat patients with COVID, and in particular how those drugs work. So thank you all for joining us. Please fill in the survey in the chat. Enjoy the rest of your day, and hope to catch up with you tomorrow. Goodbye from all of us.
Thank you. Bye-bye.